Hello, Biology 230 at Tuskegee University. This is Dr. Griffin again with video 3. Yes, we are almost there for Module 11. Now, I will be discussing DNA replication for this video 3 of 4 for Module 11. DNA replication is simply what it says. It's actually DNA making more of itself. It's replicating. It's synthesizing. So this actually takes place during the S phase of the cell cycle. Just another way for you to link some things that you've learned in previous chapters. DNA replication, both in prokaryotes and eukaryotes, is a semi-conservative process. That means that when the parent cell, here's the DNA in the parent cell, when the DNA in the parent cell replicates or copies itself, the new DNA strands, these two new double helices you see, actually have a DNA strand from the parent cell as well as a new daughter strand that makes up the new double helix that you see here. And this is how replication works, is that these two strands come apart, and then you see here in the parent cell they will come apart, and then there'll be a new strand attaching itself to this one parent strand, and then there will be a new strand attaching itself, complementary base pairing, to the other strand of the parent cell. DNA replication begins at what we call an origin of replication. And I say an origin of replication because I'm first going to discuss DNA replication in prokaryotes. Main example is bacteria. And you see here the bacteria cell E. coli. So each strand of DNA has one origin of replication. And then the parental strand or the template strand, shown in the dark blue here, they will pull apart. The two parent strands will pull apart, and then new daughter or new strands will begin to grow in complementary base pair with their respective parental or template strands. The spreading apart of the new novel daughter strands creates a replication bubble, as you can see here very nicely in this figure from your textbook. And then at the end of this bubble, wait for my mouse to come back, there begins this replication fork. And then DNA replication just continues. It just goes straight through. There's no stopping. There's no interruptions at all. In prokaryotes, it just continues. And then we make two daughter DNA molecules. And remember, you see here, the semi-conservative model. Both daughter molecules of DNA has a parental strand as well as a novel daughter strand. Now let's compare that to DNA replication in eukaryotes, in a eukaryotic cell. As you can see here, each strand of DNA in this double helix, and it's just shown linear for simplification here, but each strand of DNA has multiple origins, plural, of replication. Okay, so like before, the two parent strands or the template strands will push apart and making way for the novel DNA strands to be created. As those novel strands are being created, it will form a replication bubble, and again at the ends of these bubbles are replication forks. And this process will continue to eventually give us rise to two new daughter DNA molecules that show the same thing, this semi-conservative model of DNA replication. Now I simplified that very quickly, very easily. That's the overall concept, is that you have two parental strands, they come apart, and then two novel daughter strands are created, and then those two new separate strands, double helices, go their separate ways. Well, eukaryotic DNA replication is a bit more complex than that. Like I said, first of all, you have multiple origins of replication. Compare that to how many origins of replication per strand you have for prokaryotes. Okay? At the origin of replication, you have what we call a leading strand in one direction, where this is similar to prokaryotes, is that you will have the formation, wait for my mouse to come back, formation of a new daughter strand. It just keeps on going. It just keeps on moving, adding new nucleotides, base pair, base pair, base pair, just keeps going. However, on the other side of the origin of replication, you have what we call an lagging strand. That means there are multiple fragments that are made, multiple new fragments of DNA that are created. These fragments are called Okazaki fragments by the professor, that's PhD professor that actually discovered them, who was Dr. Okazaki. Um, Okazaki fragments, and once they're made, they then have to be combined, that I'll talk about later in the next video, to create one strand. But the main point here is that eukaryotic DNA replication is complex. It involves a leading strand where DNA polymerase is going to come in and make one smooth strand. And on the other side of the origin replication, we're going to have multiple fragments. And this strand that will be eventually formed by these multiple Okazaki fragments is called the lagging strand. Okay? So that's just a little piece. I'm going to expand on that for the next video. Please don't stop here. You just have one small part of the story. This is video 3 out of 4. You are 75% done. Please continue to learn from these videos. I look forward to discussing with you next time in class and outside of class. This is Dr. Griffin signing off for Module 11, Video 3.